Benalcazar I met as a research associate at the Chimborazo Polytechnic University. He was working in an irrigation outreach research facility, and later joined our PhD program with a Queen Elizabeth Scholarship that worked for the benefit of partner universities in countries around the world. It's funding that has its source in the Tri-Council plus the Rito Hall Foundation. Its goal is to have researchers from abroad study in Canada with an emphasis on networking and returning some of their knowledge and training to their home university. And Paul just got back from Ecuador, and he does fulfill that role of collaborating and designing new collaborations with the Chimborazo Polytechnic University. In terms of networking, Paul's been really very successful here. The Queen Elizabeth Scholarship works with internships. He worked 2 seasons with Miller Western Forest Products in Alberta. And then his 3rd internship was with the Agriculture and Agri-Food Canada station at Harrow, Ontario, near Windsor. And that helped form his PhD dissertation that he'll give to us. But there were actually 3 other outreaches that Paul did during his time. He, almost on arrival, he noticed a little posting for the Canada Agri-Food Policy Institute, which gave him additional funding for 2 years, and he collaborated with scholars across Canada, in part in writing a report on the importance of wetlands to farmland and biodiversity. He then used a MyTax Global Link scholarship for international travel and went to Cornell University, where he met Bob Schindelbeck, lead soil scientist who helped develop the Cornell framework for soil health assessment. And that's also something that really helped form his thesis. And the final bit was he funded himself to go to San Diego and worked with a colleague there who helped develop his hydrological modeling skills that he used in investing. So Paul did a lot of things while he was here that helped build the network that shaped him and brought him here today. So with that, I don't want to say more unless Randy, if you had anything to add, we'll let Paul start.
Yeah, just to add that I do quite a bit of work in South America, and usually I'm working with students that come to the States and are working in South America. I've never had the opportunity to work with a student coming from South America that's actually working in North America. So that's a big change, and I appreciate that diversity of the students learning more about forestry, American agriculture, and those things, and taking those things back to their countries as opposed to the in-country learning that I've been involved with so far. So it's been a treat.
Great. And I welcome Lori Phillips, who I just noticed joined us. She, I mentioned in my introduction, she works at the Harrow Station of Agriculture Agri-Foods Canada and can join the round of questions after the defense. I did not give credit to Randy and the OMAFRA, which is most important, Ontario Ministry of Agriculture, Food and Rural Affairs, which has a representative under bay by the name of Kendall Donahue. And Kendall, Randy, Tarluck, and Paul met on his arrival and conceived the project that Paul is about to present. So I should give credit to Kendall as well. Paul, take it away.
Thank you everyone for being here, and good morning. So today I'm going to talk about the assessment and evaluation of soil health indicators, which changed from boreal forest to agriculture in Thunder Bay, in order to complete my PhD thesis as a part of the forest science in Lakehead University. Okay, now. There can be no life without soil, and no soil without life. They have evolved together. So the importance of the study is that northern territories are part of the boreal ecosystem, and this extensive ecosystem comprises about 207 million hectares in Canada, and the current the current growing agriculture in northern regions, plus the current warming climate, has accelerated the land conversion. Changing climate has prompted policies that support land use conversion to expand food production in Canada's north, despite the limited understanding of the impact of those land conversions into soil disturbance. As soon as the soil was broken, it started to change. Therefore, negative impact on ecosystem service has been documented through those conversions. Generally, after land conversion resulted in significant losses of organic matter and the impact of soil health. When we talk about the soil health, we are talking about the measure of the ability of soils to provide service, maintain the function of the soil. Therefore, soil assessment identified the disturbance and stress that this soil has after land conversion. This dissertation covers 3 major research projects assessing the effect of forest to agricultural conversion on soil physical, chemical, and biological soil metrics in Thunderbait agricultural areas. My first research project, which is part of my chapter 2 in my thesis, determined the impact of land conversion from boreal forest to agriculture on soil health metrics. My second research project is predict the soil health indicators across gradient from newly converted to non-converted agriculture in boreal soils. My last research project assessed the impact of land conversion from boreal forest to agriculture by taking into consideration the microbial community functional capacity by qPCR and soil sequencing by operational taxonomical units. For the first research project, the hypothesis, we defined 3 hypotheses. We defined that forests will have the highest soil health score, and that clearing and conversion will result in a decline in soil health score and detrimental change in soil health indicators. Change will be greatest in plots that are more than 50 years in agriculture, and then changes will be most pronounced near to the submerged soils. For the second research project, carbon-related indicators will be the most influential of the overall soil care score in Thunderbait agricultural areas, because soil organic matter is one of the key indicators of the soil functions. For the third hypothesis, for the last project, after land conversion, functions associated with carbon cycling would decrease, while those associated with nutrient cycling would increase. As the age of the agricultural field increased, the overall abundance of fungi would decrease, while that of bacteria would increase. And finally, the overall discovery will increase with increase in land use diversity. In order to do the study, we collected samples for 2 main agricultural areas, Murillo and Slate River Valley in Thunderbait region, located around 20-30 minutes from Thunderbait. We used the split core sampler, a spherical sampler, and the sample design was 5 inner eyes, in which we use time circumvention categories, taking sample from forests at adjacent areas that has less than 10 years since conversion to agriculture, between 10 to 50 years, and more than 50 years since the conversion to agriculture. We use health depth intervals 0 to 5, 5 to 15 centimeters. We also, for the microbiology study, we use 30 grams of salt that was stored at minus 40 degrees centigrade. And for the rest of the analysis, we are dried and then we sieve, and then composite sample will send to the Cornell Soil Lab. We use a chronosequencing approach that has been used for all these 3 projects. For the first project, we used the assessment. We assessed the soil using the CASH framework, which is a comprehensive assessment of soil health developed in in Ithaca, New York, by the Cornell University, in which they take into consideration physical, chemical, and biological indicators. For the second research project, we use the minimum dataset approach, evaluating those kinds of framework, and for Thunderbird agricultural areas. For the third project, we use the qPCR and DNA protein analysis using the protocols from Agriculture and Agri-Food Canada. For the statistical analysis for the first project, we used the linear mix model, following the time series conversion, as we can see here, the depths, and the total soil sample for each category. For the second project, we use a best observed regression model that consists of all possible combinations of predicted variables, and then taking the overall soil health score. Then we use the cross-fold K equal 10 validation model that identify the best optimal model and variables that could explain what is happening in Thunderbird agricultural areas. Here we have a C category that is more than 10, less than 50 years in agriculture was added into this study. The total number of samples for each category. In addition, for the analysis, we merge all the plots that are related with agriculture, and then we merge all data sets that include the forests. In terms of the third project, we use for the first part the microbial community and functions by qPCR. We use the one-way ANOVA and Kruskal-Wallis test according with the distribution of the data. The time series conversion we already mentioned, the depth. By the second part of the analysis sequencing microbial community, we analyze at the class level the fungal and bacterial communities, which relative abundance is more than 0.05%. And then we use the non-parametric multidimensional scaling with Permanova. And then for this microbial community analysis, we use the vegan package in our software. For the Bright-Curtis dissimilarity index, Adanis functions that is part of the vegan package, and the Shannon index, and also we use even the ANOVA or the Kruskal-Wallis test. For the first project, it's important to mention here this is already published in the Canadian Journal of Soil Science. And what we did discover, so the land conversion resulting in degradation of the soil health, and those changes were especially this degradation especially was found at surface about 0 to 5 centimeters that occurred during the first year since the conversion to agriculture. We can say, for example, here that biological and physical indicators were the most show significant differences through the conversion. Here, for example, organic matter have decreased at 0 to 5 centimeters depth by around 48%. Then, for example, soil respiration have declined since the conversion to agriculture around 25%. And wet aggregate stability, that one of the physical components, have declined around 27%. Then following the result, also we found that total carbon and total nitrogen have declined since the conversion and has shown significant differences, especially with 0 to 5 centimeters. Here, for example, phosphorus has increased around 59% in the conversion, and active carbon have declined, decreased around 20% since the conversion. Then for the overall soil health score, that as we can see here is calculated and scaled for from 0 to 100. We can say that forests, it's very high, that it's comprised, take into consideration values that are more than 85%, so those are very high overall soil health score, and between 70 to 85 is considered to high overall soil health score according with the CASH framework. So for agricultural side we have 79%. Moving to the second project, we used the best subsurface regression model that help us to identify what is the best predictor for a single or two variables or three variables, and what are the most influential of the overall soil health score. For example, here we can see that most of the indicators are related with the carbon, influenced by carbon. And for example, let's see, for example, when we combine all the plots that are related with agriculture, we can say that respiration is the best predictor for a single variable with R square of 0.46. And when we take into consideration two indicators, we can say, for example, that the compaction surface hardness and respiration are the best predictors with 0.57% R square. And taking into consideration three variables, we can say, for example, surface hardness, respiration, and pH are the most important best predictors for Thunder Bay agricultural areas. Moving to the cross-fault validation approach, the optimal best model for Thunder Bay agricultural areas, we can see, for example, organic matter, active carbon, protein, respiration, aggregate stability, hardness at 0, 15 and 15 to 30 centimeters plus manganesium are the best indicator that describe what is happening to the soil since the conversion, taking into consideration functions not only related with the nitrogen cycle, soil resistance, soil resilient, water cycling, carbon sequestration, but also habitat provision and human health. That will help not only with the actual managed practice that we have right now, but also the current climate change that we are facing. For the third project, the assessing the microbial community functional capacity by qPCR and soil sequences by operational taxonomical units, we can say that agricultural soil in Thunder Bay areas have increased relative abundance and total archaea fungi compared to forest systems. As we can see here, for example, archaea and fungi has significant differences since the conversion to agriculture and have increased about 64%, 36%, although bacteria relative abundance doesn't show any significant difference. The relative abundance associated with change related with carbon cycle, we can say that complex organic matter decomposition like acid did not change a significant change since light conversion. However, the relative abundance of the composition of low molecular weight carbon compounds have increased, as we can see here and here, and also in the next slide, this one that have increased 33%, sorry, and those are also related with the decrease of soil organic matter and increase of phosphorus and potassium. Then moving to the relative abundance of genes associated with nitrification, we have shown, for example, here a significant increase following land conversion, especially for these three genes associated with nitrification. However, those associated with denitrification, as you can see, -1, all this -1%, doesn't show any impact since the conversion to agriculture. Moving to the relative abundance of genes associated with phosphorus mineralization, we can say the phosphorus C and phosphor D, acid and alkaline phosphotatsam, decreased since the conversion to agriculture, and especially due to the input of phosphorus that had been into place in order to produce food. In addition, looking at the channel diversity at bacterial, we can say that the bacterial diversity has increased since the conversion from forest to agriculture at both depth, as you can see, 0 to 5 to 15 centimeters. However, there are no significant changes in the fungal community at both depth as well. The overall bacterial fungal diversity evaluated by sequences also changed after land conversion. As you can see, according with time since conversion, we have this fungal diversity also moving along for bacteria and fungal microorganisms. Moving to the operational taxonomic units and the bacterial taxonomic distribution from time since conversion, it's important to mention that Firmi, Cutes, and Hematinoma tudetes have increased since the conversion. On the other hand, Actinomycetes, Acidobacteria, and Protobacteria have decreased. It's important also to mention that functions associated with these bacterial organisms are related with carbon cycling, little decomposition, carbon and nitrogen recycling, nitrogen fixation, and nitrogen provision. For the fungal taxonomical distribution for time since conversion, we can see the Ascomycota has increased, while Basidiomycota have decreased. And especially Ascomycota have increased due to the resilience to the managed practice that we have found in Thunder Bay agricultural areas. For the first project, we can say that land conversion resulted in soil health degradation, but those changes especially happen in the first 0 to 5 centimeters and in plots that are less than 10 years since agriculture. And the loss of organic matter in Canada is around between 15 to 52%. And this loss of organic matter is due to declines above and below ground vegetation, where dead roots are the primary source of carbon. In addition, with farm practice like tillage that increase and also the increase of temperature that expose the soil and accelerate carbon decomposition. For the first research project, it's important to mention also that in Thunder Bay agricultural areas we have high levels of soil organic matter concentration. That we're talking about the 100 between 114 to 69 milligrams per gram, while in southern Ontario we are talking about an average values of 43 to 16 milligrams per gram. Therefore, these give us as a conclusion that why Thunder Bay have high levels of overall soil health score. And this is due to the soil forming factors. This high level also is important to mention that we are using the cache framework that was developed for the United States, especially in New York. So the climate condition and forming factors are completely different what we have in Thunder Bay. For the second study, we can say that when we take into consideration the indicators that we are describing right now, we have to take climate, parental material, sectoral class, and management practice. Pay attention to those factors. Then also, as we know, boreal soil have a high levels of organic matter, as I mentioned, not only due to the low temperature and reducing microbial decomposition. Then talking about the minimum data set that we identified for Thunder Bay agricultural areas, we can say, for example, that soil organic matter is a key indicator to describe what is happening in terms of physical, chemical, and biological components of the soil. When we look at for soil respiration, ACE protein, and active carbon, those biological metrics are usually used to detect seasonal change. Also, it's important to mention that due to this current warming climate condition, rates of soil organic matter turnover and mineralization will be important to monitor, especially in Thunder Bay agricultural areas. Then for soil aggregate stability, is an indicator that provide a stage of what is happening with carbon sequestration. When we talk about soil compaction and soil hardness at about depth, we are talking about the movement of the water, the soil infiltration, and therefore with the increased precipitation also it is important to monitor those changes. Finally, magnesium can be used to determine carbon stock. For the last project, we can say the land conversion impact abundance and composition of microbial communities. Agricultural practice like tillage, organic matter addition, crop rotation, crop resistance influence the microbial community abundance and composition and activity. Relative abundance of archaeas have increased the nitrification process on the soil, and also nitrification with amino A has positive response in acid soil, and also the tillage, liming, and cattle manure application has positive effects of nitrification. Lower molecular weight carbon have increased, especially with cellulose, hemicellulose, and sugar that are part of the materials after harvest a crop, while soil organic or marine matter decrease and potassium and phosphorus increase. The bacteria diversity have increased and by management practice, tillage, but have reduced the fungal diversity. For the bacterial abundance have been reduced due to the application of wood, ash, and liming in acidobacteria, actinobacteria, and protobacteria. To conclude my studies, for the first project we can say that physical and biological indicators are the best predictors in identifying what is happening into this soil. Land conversion has followed it by declines in weather aggregate stability, soil respiration, active carbon, and concentration of organic matter. For the second project, indicators associated with soil function related with nutrients cycling, carbon sequestration, habitat provision, water quality, climate control, and crop production were the best predictors for Thunder Bay agricultural areas since the conversion from forest to agriculture. And then for the last project, a new agroecosystem has that replaced forest, enhanced carbon and nutrient mineralization and decomposition of organic matter. But after just 10 years old of land conversion, change occurred to microorganisms abundance and diversities, and while the composition continued to stabilize. As a general conclusion for the whole research project, we can say that soil health assessment and evaluation is an integrated approach that helps us to recognize what is happening to the soil by looking at physical, chemical, biological indicators. Also, this comprehensive assessment of soil health, along with a quantitative polymerase chain reaction and genes sequencing, help us to detect what is happening into the soil since the conversion. And here also is important to mention that in Thunderbird agricultural areas predominantly are dairy farms that grow forage crops and cash providing insight into those soil degradation, especially for the first 10 years in the conversion. However, this continued amendment of the application of manure show that after 50 years of synthetic conversion, the soil has started to recover. Finally, we can say that due to this climate condition, specific characteristics that we have here in the north, plus the actual picture that we have right now in terms of the assessment and evaluation of this soil health, and the biological indicators that have increased after 50 years in agriculture, we can say that agriculture in the north with dairy farm hasn't impacted the soil functions in the soil, and probably talking about the future and the policy, we can say that northern areas agriculture could be focused on those dairy farm, according with what we have related with the manure application, the cropping system that we have right now, and also the rotation that we have, plus adding the conditions in northern Ontario, especially the frost-free season and the long winter periods that more or less help soil to recover. At the end of my study also, we can say that for the project 1, the hypothesis 1 is true, the hypothesis 2 is false, the hypothesis 3 is true. For the project 2, we can say that the hypothesis is true, and the project 3, all the hypotheses were true. Finally, I would like to thank all my committee members, research collaborators, and also the institution, organization, and funders that were available in order to complete my study. And thanks for all of you for those hours and hours and guide and help. Thank you so much. That's all.